All right. Good morning again. So this is our second session on this topic called modeling the neural syntax. And we call it neural syntax because so far only we can understand. Our understanding has to go to how we speak. I'm speaking now. So this speech that I'm doing is coming through many operations in the brain. And then as it reaches you, you are trying to handle it with many operations in your brain. So I'm a transmitter and you're receiving it. Now, you are not expecting what I'm speaking, but you are able to grasp it. Now, people say you're able to grasp it because I'm speaking in a language that you understand. But that is where the fallacy is. See, it is not the words that you understand. The words going into you become electrical signals. Those electrical signals transmit through. So then they reach the brain, and then they become from electromagnetic operations to mass operations, thermodynamic operations, balance operations, equilibrium operations. So through that, so there are events in the brain that will detect that this part was communicated, this part was communicated, this part was communicated. Now, those communications, probably you have some in the memory before, which are invoked, saying that, is he saying the same thing as before? So that helps to create a bandwidth to say, do I listen to it or do I reject it? Have I heard before? So that whole process is the audio reception interpretation that we want to study. So today, so last time, our friend Bharani Padmanabhan, um, he presented 2 months ago on this reception of the audio signals. What we said that the reception of audio signal is instantaneous. So that has some kind of a response that we can all experiment, that the nature of this signal is understood by a protein rather quickly to alert you what's coming. Now, today what we'll do is, through that mode, which basically means that you have created a mental envelope where the stimulus has been alerted to you electronically, alerted to you that this is Bijoy, he's a friend, he won't hurt you, or this is a tiger, I'm in danger. So that communication has taken place. After the communication takes place, what does the brain do? So this communication takes place to a limb called thalamus. So from thalamus onwards is what we'll understand today. To speak to us is Mr. Prem. By the way, before I do, let me acknowledge friend Bharani Padmanabhan. We are a 4-member team here. Bharani Padmanabhan and our friend Dr. Bela Kosaras. So they both are Bela is a neuroanatomist, Bharani is a neuroscientist, and I'm a physicist. And Prem Nagar will talk today. He's a computer scientist and biologist. So we are welcome new people also who want to join us. So let us hear from Prem. So Prem's topic is reading the neural centers. So neural centers are these thousands or millions of little tokens which are connecting to my speech in your brain. So my words have been converted to electromagnetic signals. They are in the brain now. So that electromagnetic signal has to be patched. English has to be obtained from it. There's no English in the brain. Brain has a neural signal. So that part we'll try to understand in our way today. So Prem is a biologist, as I said. He has been doing computer science for a long time as a database manager and then retired. And so let us hear. Now, the questions that you may have, hold them. We'll have enough discussion. You can, and also we want to learn from your own empirical observation. We want to learn. So do share them. That this is what happens. What we're doing is we're trying to create a physical framework, how the system probably functions. It is not really total because, as you can say, we're defensive. I mean, it's the human experiments are not done, so we have to interpret them from our own basic empirical observations as things happen to us. Here he goes, Prem Nagar.
Let me share the presentation. Thank you, Dr. Bijoy Misra, and good morning and welcome you all for this session. My name is Prem Nagar, and I'll be discussing today about the reading the neural centers under the broad topic of modeling neural syntax. I acknowledge Dr. Bijoy Misra and Dr. Bela Kosaras as my I start. Now, coming to the topic, modeling neural centers or neural syntax and reading the neural centers. Neural syntax is a way to organize patterns into a meaningful unit that can be processed. Modeling of the neural syntax essentially relates to the brain processes. The neural centers are a group of neurons that are organized and specialized for a particular task. These centers are integral to brain functions and critical to our cognitive, sensory, and motor processes. Today, in the reading of neural centers, we will discuss the constituents of these neural objects, their functions, and their interplay as we see it. Now, modeling the neural syntax for an audio signal, we propose a computational model to determine the neural syntax as a precursor to speech. Now, this model has 4 parts: reception of the audio signal, and this was done by Dr. Bharani Padmanabhan on 21st of October, a couple of months ago. Interpretation of the audio signal, which is reading the neural centers, is being done today. And the concept formation, the development of neural clusters, and neural syntax, the Paninian analysis of human cognition, these two will be done in future by Dr. Bela Kosaras and Dr. Bijoy Misra. The model is, we receive the audio signal, we interpret those signals, we organize these signals, and then we express those signals through speech. Now, before I get down to the reading of neural syntax or neural centers, let's recap the reception of audio signal, which was done sometime back. The real-world empirical data suggest that the speed of audio signal reception is much faster than it is suggested by the mechanical models of nerve transmission. Because as I speak, you are able to get it, understand it. So there is an instantaneous communication which we have. Now, this has been shown already in the case of reception of light signal by retina and nose. We thus propose that we apply the principle and techniques of quantum mechanics instead to model the reception of audio signal as well. Empirical observations, the emotion is instantaneous, which is in the femtoseconds. Now, in this new paradigm, the quantum tunneling of electrons, we thus propose the essence of proteins in the cochlear hair cell and suggest a rapid electromagnetic 3D surface ring plasmon polaritone wave transmission in the XML space, differing from the ion movement in the classical model. We propose the presence of receptor protein with femtosecond electronic change, suggesting a real-time processing at the femtosecond speed in neural transmission. The model proposes segmented audio parsing right at the cochlea itself. Now, from the cochlea, there are potentially 30,000 auditory nerves which carry this electronic signal, activating neurons, protein at the auditory cortex, forming a neural assembly that are processed through reader neurons. Now, the present task, the interpretation of the audio signal, as posited, the auditory signal is adaptively encoded with regard to pitch, tone, loudness, and emotional timbre, forming part one of the model. Now, subsequently, the audio signal undergoes interpretations by activated neural centers responding to the initial indication of emotion. The present goal is to understand the processing of the audio signals by these neural centers and decode them using the reader neuron infrastructure. Neural centers are shaped by the experiential learning in each human. Human interpretation inherently subjective and is colored by their memory. Neural centers influenced by genetics, experiences, environment, and undergo dynamic interplay and continuous adaptation and refinements of the circuits throughout individuals' lifespan. Now, coming to the neural assembly, this is a group of interconnected neurons that collaborate actively to process information, enabling the formation and storage of memories and facilitating various cognitive functions in the brain. Now, if one looks at the current traditional auditory information flow, the cochlea, which is in the inner ear of the humans, converts or transduces sound waves coming to our ears as the mechanical vibrations into neural signals through hair cells. Neural signals travel via auditory nerve to the brainstem, where basic processing occurs. Processed auditory information ascends to the thalamus and auditory cortex, enabling complex perception and interpretation of sounds. Now, neural assembly, we hypothesize that the attributes of these auditory stimulus form a neural object called cell assembly. A cell assembly is a group of neurons that function collectively to represent a neural object. The concept of cell assemblies suggests that such a group of neurons work together to encode a specific feature or an attribute of a particular object or a sound object in this particular case. Together, these attributes as cell assemblies contribute to the auditory experience that we have. Their representation in the brain is the coordinated activity of neurons across auditory pathway that forms a neural assembly. Now, this is a traditional auditory information flow, and I'm showing this as a sequential, but it is not sequential, it's a nonlinear. The first box is the cochlear nuclei, which is in our ear, where the transduction of the sound to electronic signal happens. So maintaining a for pitch and frequency interpretation up to the auditory cortex. Now, the auditory nerve carries cochlear impulses to cochlear nucleus, which is divided into dorsal and ventral segments and projected to superior olivary complex and lateral lemniscus. Now, superior olivary nuclei contains medial, lateral, and medial nucleus of the trapezoid body, sends projections to lateral lemniscus. Lateral lemniscus gathers input from various auditory nuclei and relays information to the inferior colliculus, and inferior colliculus projects to the medial geniculate nuclei, the MGN, which is the relay center in the thalamus, that acts as a relay between the inferior colliculus and auditory cortex, and it filters and processes various sound features before transmitting to the cortical areas. The primary auditory cortex, which is A1, interprets basic sound characteristics like pitch, loudness, and duration, aiding in tonal respect. The secondary auditory cortex, which is A2, discerns sound patterns and extracts detailed information from auditory stimuli. And the adjoining area, which is the association area, attribute the meaning to the sound and link auditory stimuli with memories and emotions. In parallel, there is a limbic system, hippocampus and amygdala, that contribute to the auditory processing and memory. Amygdala processes emotional significance attached to the auditory stimuli, forming emotional memories and triggering emotional response like fear, pleasure, or excitement. Hippocampus gathers information from sensory areas and auditory cortex. This is a key for encoding and consolidating a declarative memories, their facts, events, and experiences. Now, coming to the neural centers. Now, brain is a collection of distinct modules grouped forming a specific function rather than unified system. Brain is an electromagnetic ball composed of neural components to have the continuous communications with one another with alpha, beta, gamma, and delta waves, and gets produced spontaneously in this self-organized manner. Now let's explain, or let's understand the constituents of the neural elements and their play to present the process that we suggest. So in the recent Human Brain Project, which was funded by Obama's presidency, that discovered more than 3,000 types of human brains. The primary functional cells are neurons, astrocytes, and glial cells. All sensations, movements, thoughts, memories, and feelings are the result of the signal that pass through these neurons. Now, the picture of the neuron is shown here. This is a cell body. There are the dendrites. This is the axon, and these are the axon terminals. And this is the direction of the signal. So the neuron consists of three parts: the cell body, dendrites, and axon. And if you look at the images here, these are some of those new cells which they have found. And these are the images of those cells. Now, what is the neural center? Now, neural center is a group of neurons in the specific brain regions that are organized for a specific function. Now, this is a picture here for a neural center. It's a complex network of neurons. Now, these centers could be localized structure or distributed networks for processing and integrating information. They serve as a focal point for information processing and integration within the brain. Now, second image is a neural circuits. So neural circuits are interconnected networks of neurons that serve specific functions involved in the communication between various neural objects. Now, if you see here, there are three different types of neural objects: sensory neurons, interneurons, which are connecting, and motor neurons. So these are three different types of neurons. Oh, one. Circuits may span multiple brain regions and play critical roles in the execution of cognitive tasks. Now, coming to the brain oscillations, which are the results from rhythmic neural activities that balance between excitation and inhibition within a neuron. So the neuron membrane inside and outside, what the plus and minus voltages are, that determines that excitation or inhibition of the neuron. And there are two images here: neuronal spiking at the top and local field potentiation, and we'll get to the details of it. The oscillations coordinate neuron group for information transmission to process incoming signals during both the sending and receiving phase. Their significance lies in establishing timing for fundamental functions like memory and perception. And permanent brain oscillations are consistent rhythmic activities for neural communication. Now, coming back to these images, if you look at the simulation of neural oscillation at 10 Hertz, the upper panel shows the spiking individual neurons. Now, each dot representing a particular action potential. And the lower panel is the local field potentiation reflecting their summed up activity. Now, coming to the plasticity, that how we adapt. So the neuroplasticity reflects neurons and neural networks inherent to modify interconnections and behaviors in response to new information. Now, this adaptive process involves adjusting both the strength and quantity of synaptic connections between neurons in the brain. It's interesting to note that in the cerebral cortex, neurons start with around 2,500 synapses in the embryo, which increases to about 15,000 synapses during infancy. So the diverse plasticity forms, including long-term potentiation for stronger synapse to spike timing-dependent plasticity, crucial for learning, and homeostatic plasticity for stable neural activity that enable a rapid adjustment and cognitive flexibility. Coming to the signal interpretation by neural centers. So in this new paradigm, the auditory signal interpretation involves the coordination between cell assemblies and reader neurons. These cell assemblies, fundamental to brain function and can trigger specific response to downstream neurons without external reference. The evidence supports collective, selective, and flexible reader activation implementing pattern separation and completion at the membrane integration, synaptic plasticity, and gamma oscillation timescale. Now, assembly reader coupling exhibit plasticity, selectivity, associative learning, and a reader-centric framework proposes characterizing cell assemblies by their ability to elicit response in target neurons, revealing a bidirectional communication. So how the reader neurons assumes the characteristic of the assembly, that is how it is described. The computational properties demonstrate assembly reader relations that undergo selective changes during learning and memory processing, highlighting the dynamic role of cell assemblies and auditory signal processing. Now, we propose that reader neurons situated in the hippocampus are proteins, which are metastable proteins, capable of receiving electrical packages, information from neural centers to establish connections with memory. The interrogation process for memory involves recognizing pre-existing patterns encoded with the data. Pattern recognition is accomplished with remarkable efficiency, requiring only a limited number of data points. Memory system is adept at recording new and unusual patterns, treating them as novel signatures. Understanding the intricate mechanism of memory interrogation and pattern recognition in the hippocampus contributes to unraveling the complexities of the neural information processing. Now, cell assemblies and the reader neuron as fundamental units of cognitive brain activity. They introduces reader-centric framework where cell assemblies are defined by their ability to trigger specific response in the downstream neurons. The downstream neurons has to assume the characteristic of the cell assemblies. Experimental evidence obtained through large-scale neural ensemble recording supports the existence of assembly reader communication. Results indicate that activation of cell assemblies precedes and influences downstream reader neurons, demonstrating collective, selective, and flexible communication. Assembly reader mechanism exhibit both pattern completion and pattern separation properties evidenced by reader response to partial assembly activation with observed changes in assembly-reader relations on fear conditioning. It thus reveals changes in assembly-reader relations during associative learning, emphasizing the plasticity and behavioral relevance of this mechanism in the brain function. Now, coming to this interactions of hippocampus, thalamus, and cerebellum, where there is an interrogation also happening. It's a very simplified diagram which illustrates the functional connectivity. The top memory, hippocampus, at the bottom cerebellum, and this is the auditory stimulus which we are receiving through our ear and going through various nuclei, and it reaches to thalamus, which is a relay center. So thalamus serves as the relay station, receiving auditory signals from ears and relaying them to other brain regions. Hippocampus is crucial for memory and spatial navigation, contributes to the perceptual aspects of the audio signal processing. Collaborating with the neocortex, hippocampus contextualize auditory information, linking it to memory experience. This is the area. Cerebellum, traditionally associated with motor functions, plays a role in the timing and coordination aspects of the auditory task. Neocortex is vital for cognitive function, processes auditory stimuli, and collaborates primary auditory center, the neocortex, analyze diverse auditory stimuli, contributing to the perception, and cell assemblies in these regions exhibit flexible communication, influencing downstream reader neuron. Hippocampus, thalamus, cerebellum collectively contribute to the bidirectional communication and dynamic processing of audio signal, highlighting their integral role in auditory perception and cognition. Now, let's get to the details of some of these aspects. So hippocampus play a vital role for memory and links with the neocortex to process the auditory signals. Thalamus relays auditory input to the neocortex and amygdala for diverse processing. Amygdala, which is a central to the emotions, assesses emotional significance of the auditory stimuli. Cerebellum, tied to the motor control, contribute to the timing in auditory tasks. Interactions among hippocampus, neocortex, thalamus, amygdala, and cerebellum form a complex network for auditory processing. Hippocampus and neocortex integrate auditory data with memory and context. Thalamus serves as a pivotal relay for auditory signals, influencing diverse cognitive and emotional response. Understanding these interactions illuminates multisensory auditory processing and its links to memory, emotion, and motor coordination. Now, how the memory interrogation mechanism works. So cell assemblies are recognized as the fundamental units of cognitive aspect. The communication through reader neuron can potentially evoke response in the downstream neurons. A reader-centric framework has the capability to elicit specific response in the downstream reader neurons. Cell assemblies reliably induce spiking in downstream neurons, demonstrating a dependency on the collective activation. Assembly reader mechanism exhibit both pattern completion and pattern separation properties as evidenced by reader response to the partial assembly activation with observed. Now, how the neural center, thalamus, and hippocampus does the interrogation. So the neural centers, biologically active cognitive tokens, are linked to the thalamus to receive stimuli information. Neurons convey stimulus signature as a form of energy transfer to the thalamus, perturbing metabolic processes and generating heat. Neural centers absorb excess heat, leading to the creation of metastable proteins. Hippocampus detects these perturbations, subsequently activating the corresponding neural centers. Engaging in a dialogue with memory, hippocampus facilitates pattern recognition, with neural centers serving as a token defined by its relationship to the motor control. Cerebellum and neocortex processing of auditory stimulus. In case of cerebellum, they contribute to the timing and coordination aspect of the auditory task. Its involvement suggests an influence on the motor response in response to the auditory stimuli. Neocortex is vital for the higher cognitive functions. They process auditory stimuli from the thalamus and collaborate with hippocampus for contextualization. As a primary auditory center, it analyzes diverse stimuli, contributing to perception, understanding through intricate neural circuits. Now, motor activation mediated by thalamus, hippocampus, and amygdala, the hand and the gestures which we see on our face or on the listener's face. Our hypothesis posits two motor responses pathway to auditory system. A semi-conscious route involving the thalamus, hippocampus, amygdala, and conscious route through the prefrontal cortex, influencing the motor actions via the motor cortex. The amygdala evaluates emotional significance in auditory cues, impacting the motor action with connections to the prefrontal cortex and motor planning regions, influencing responses. Investigating these pathways reveals the interplay between sensory processing, emotional assessment, and motor response. Now we come to the proteins. Now proteins are biopolymeric structure composed of amino acids, of which there are 20 commonly found in biological chemistry. Proteins serve as structural support, catalyst, hormones, enzymes, building blocks, and also, interestingly, initiator of the cellular death. Proteins synthesized from specific genes to serve a distinct function. Now, the protein exhibit structural elements such as helix, strings, and sheets. The regulatory role of metals, including iron, zinc, and magnesium, etc., modulates the protein function. Human Protein Project, this is one of the United States project, identified protein expressions for 93% of the 19,750 predicted proteins encoded in the human genome. Now, proteins prevalent in the human brain include neurofilament, GFAP, MBP, and others. Understanding gene-coded proteins structure, metal regulation advances proteomics. Now, here is the image of the protein. that consist of 3 helix, helix 1, which is in red, helix 2, which is gray, helix 3, which is blue, connected by 2 turns, turn 1 and turn 2 respectively. Now, protein structure and process of reconfiguration. The protein's definitive structure is determined by its amino acid sequence, featuring 4 hierarchical levels. Primary, secondary, which is a local folding into alpha helix or beta sheets. Tertiary, which is overall 3-dimensional form. And quaternary, the arrangements of multiple subunits. Now, protein influenced by their environment undergo conformational changes, including interactions with the metals. Now, this dynamic configuration or reconfiguration, driven by factors like temperature, pH, that contributes to the diverse proteins in the human protein, categorized based on function, structure, and properties. Now, how the proteins signal each other. So this is the protein signaling process. So protein engage in communication through chemical modification. And these are the 2 chemical modifications, which is given here. A phenomenon known as protein signaling. So it's more of a chemical modification, that is the protein signaling. Upon modification, proteins undergo property changes, transitioning between inactive and active states or modifying their interactions with other proteins. Now, this is an inactive, I'm looking at the images. This is an inactive protein image. This is one of the protein which is in some kind of chemical process. And these are 2 different proteins. One second, sorry, yeah. These are 2 different proteins which are in the communication. So the protein variant. So cells describe DNA to RNA, splicing exon into mRNA, creating varied proteins with their distinct functions. The graphical annotation illustrates protein structure influenced by habitat, affecting their configuration in different environment. So this is the DNA, I'm looking at the image. This is the DNA, which is expressed in RNA. Then there is a splicing, mRNA. These are 3 different translations, and which has created protein A, protein B, and protein C. So this the overall process, and protein stability in a cell milieu is a function of its electric field and local environment. Now, the state of protein or the metastability. The protein stable state is defined by its environment and what environment it is. The environment changes with the external activity and metabolism. Metastable states are assumed to be Markovian, only dependent on the previous state, not a memory. Now, these are the 12 different states of the protein. It's a molecular structure of 12 highest populated metastable states of HP35. And this consists of 3 helicals, 1, 2, and 3. And they're connected with 2 turns, turn 1 and turn 2, respectively. And there are 12 such examples. Now, coming to the summary of this, a cochlear hair cell's protein transduces sound vibrations into audio signal that travels at the electronic speed of femtosecond. Now, through assembly, reader mechanism, the neural centers, a biologically active cognitive token, dialogues with memory. Neural centers specialize for coded information, decode audio signals through protein, possibly through their metastable states. Signal processing in the brain involves neurons providing a subjective interpretations. Our hypothesis suggests decoding occurs through protein reconfiguration within neuron interrogated by the reader neuron. The specifics include the number and nature of protein in the processing remains unknown. Now, the concept of neural object, the neural assembly, neural centers, reader neuron, and others shift focus from individual neurons or brain regions to interconnected patterns of neural activity, emphasizing the dynamic nature of information processing, and these objects seems to be shaping the perception and cognition. Studying their creation, structure, and manipulation may yield insight into the intricate brain functions. Thank you.
Can you all unmute yourself and have a round of applause for Prem?
Very good job, Premji.
Thank you.
So, okay, so before I think let us discuss it. I think an important point that has to be communicated, the difference between animals and men, people. It seems we, each of you, is more action-oriented. Each of these neurocenters, that slide did not come up, each of these neurocenters is connected to the brainstem. So they have not only they're receiving information, stimulus, like I'm talking to you, so that is coming to you. At the same time, you are reacting. What do I do with it? It is not understanding of it. It is not the location of it. It is the ownership of it. I have to do something with it. So that something with it is very human. So that is where we have all this brain functions saying, so the kind of divisions it must make to say what actions are possible. That is for example, I'm moving my hand here, my degrees of freedom of my body. So all those are degrees of freedom, my muscles, my veins, my neurons, my eyes. So those are all aspects of way the body is trying to find a response to the stimulus. Actions. And this is philosophical. Jaspal is here and so on. It's philosophical in the sense that we are not understanding oriented. We are action oriented. And that somehow in our model, we think that is necessary because stimulus brings energy. So that energy needs dissipation, has to go away. At the same time, the action is personal. That is, action is not generic. So every individual creates its own threshold that how much, what they will receive, where they will act, and what they will act. So these neurocenters are operating in that plane. They have developed themselves through the human habitat, our own functioning, our parents, our culture, our locality, the way we operate, and so on. So they have created a sense what to expect from the external world. The sun will rise, air will come, trees will form. So we are not in a desert planet. So like that. So they have an expectation. So that expectation in the neural centers has been coded through these long years of habitat. Now, memory forms through these processes you store, but memory is more organic than local. A tree is a tree in the memory. It's not my tree or your tree or his tree. So there is a general reception of the object that the memory etches so that the object is identified as the object, not the way I interpret the object. So there's the object universe, which is etched in the. So this is the model. So memory in the human memory. So that when I call a flower, you have a flower, things like that. But then your action when I call a flower is different. So that is where the hippocampus comes in, where the neurocenter is communicating the hippocampus that this Bijoy Misra is saying flower. So the hippocampus then asks the memory to say, is this Bijoy Misra fellow important? Then memory says, no, ignore it. Oh, no, I have seen him before. Oh, he says flower. Oh, flower. Okay, what flower? Is it winter? Is it a flower? So those kind of communication come from the memory to interrogation that is it something I should know about, which is basically entropic information increase. That is, memory knows it's winter and so on, and there's a flower now, so there is a new information comes in. Memory alerts. So that dialogue, so through hippocampus, it says, okay, what kind of flower are you talking about? Paper flower, glass flower, regular flower, forest flower, what is this? So then hippocampus, the neural centers then furnish information. The red flower, oh really? Indoor flower, oh okay, all right, things like that. So that kind of a larger picture that we are trying to understand. So this action on the brain stem is where our expression comes in. That is, some of the original actions could be pretty nominal, movement, shivering, breath, vessel, dilation, maybe a little muscular on the face. But eventually, if it really builds up, and if you feel like, then you may get some grunts. In higher level, if you hold the object inside of you, you may try to express in syllables. Why syllables? So this is where other physics comes in. Syllables are our, in the Paninian model, the way he developed it, that is, we are the human beings. We are syllabic. Through our transmission processes of air in the body, we are tuned that the following phonemes may result. It's not our choice. That is how the body is designed. The throat, the vocal cord, the nose, the teeth, the tongue. So they have the manipulation of them will produce, which basically then means that whatever the object is, whatever the stimulus is, whatever the scene may be, it has to be channelized if to be expressed acoustically in this Paninian syllables. They're called Paninian, but basically human syllables, which are discrete. Now you can combine them and so on. That's a different kind of physics. But what that boils down to is that each syllable in that sense does represent some part of the universe. Some part of the universe. So anyway, so we'll come to that. So I just thought I throw this away for this action part of it, that the neurocentres are connected to hippocampus, also connected to brainstem. So through brainstem, they're dissipating. At the same time, they are kind of letting action form. We have not studied how much of when let us say the dog runs away, the insect moves, that's also action. But our actions are much more, degrees of freedom are much larger. And then on speech, it's extremely intricate. There's a context, there's a tone, there's emotion, there's a value, reason. Then also there's prosody. So we'll come to that gradually. Just building framework. So ask any question that you have, and then examine and understand, or help us understand how we speak or why we speak. Any questions? Welcome.
Can I ask a simple question?
Yeah,
I have a simple question.
Yeah, please.
Is this model, network model, brain, is it only for acoustic impulse?
No,
what
no,
about
no.
It is very general. It looks like because
right. No, we take acoustics to be to just simple.
It's our model, but the whole brain structure,
right.
neural centers, and
right.
they're
No,
very
more. I think they are more. I think we do not know. We're studying. We do not know. So the year, what happens is linear. So it's going. So where we have a pathway you can find. That is why we choose the audio, in the sense that the integration of it. You are receiving it as a time sequence, and that time sequence eventually gets integrated. But rest of the senses, they are not time sequence, they're kind of loading on you.
Because it took very general concepts.
Right. No, I think this is probably modeling point of view. We do not know, though, what is the time increment one may do, and that we are studying through your, for example, the way we understand this process, that itself is a parameter. Cognition is a function of how quickly you register a stimulus, and then how much of that is due to habitat, and how much is that due to strangeness. So if you make a model or philosophical model that the human brain is capable of handling any stimulus. That is, suppose you throw in a galaxy, you don't never been there, but you can handle it. What is it? So that the brain has that ability to take an object, any object, and then interpret it. So those interpretation, the question is, how does brain do that? So the question then would be, naturally, it is not infinite process. You have only finite time in order to process it.
And also finite time to respond and finite time to come.
So that basically means that the object somehow has to be segmented into the brain channels, whatever the object is. Each of it is an object. The sun is an object, galaxy is an object, interaction is an object. So that has some kind of a way.
I'd like to follow on with this question. Is it acoustic or is it optical or something else? For example, I'll tell you what happened to me right now is that my brain told me that I'm missing something. And what was I missing? I'm missing the 9 a.m. Zoom meeting of Prem Nagar's, So how did that happen? I did not get a voice. There was no lighting that came to my mind. So that's my question. So whatever my brain is capable of telling me you are late, but still there may be some time that you can go in. But there was the fact that there was a event that I'm supposed to attend, and I did not. My question
Right.
is that
I think
what
those
sort
are
of stimulus,
clinical.
what sort of stimulus
Right. We have to
the
distinguish
brain
between the
uses to
Right.
get that.
Right. The
Maybe
clinical
we can go
thing.
ahead, and if we don't get into Bijoy, I think you are dismissing this
No,
is
I'm not
something
dismissing.
not
I'm not
very good
dismissing.
because I'm not talking to you. I'm talking to the entire team. Okay. So the point I'm making is very important because the question that Professor Mahanti asked is an extremely important question. Is it only acoustic signal?
No.
Is it other signal? And if it is not acoustic signal, are we doing a mistake by concentrating on acoustic waves? Because acoustic waves are very specific. They don't exist. They can be blocked. And that's the reason I tell you that
I'm not
assumption. You
I'm
can
not
go.
disrupting.
No, let me finish. I mean, I'm not addressing to you. I'm addressing.
I'm not. But introduce yourself.
You can answer. You can answer after I complete my, my point. The point is simple. Okay, what is the brain? How does the brain function? It is complex, but we should not. My point of view is that we should not limit ourselves by using a model, by using a acoustic model, which is very specific. Okay, thank you.
Thank you, Devaji. Let me handle Bijoy. Give me 1 minute.
Yeah, thank you.
Devaji, yesterday I had an appointment for my eye test, annual appointment. And I was emphasizing the doctor that, listen, why I'm not able to see perfectly well, which I was able to see some time back. He gave me a good answer. I mean, I think it's a good answer. That he said, supposed to have 180 million neurons, or not neurons, but the optical receptors or photon receptors to see clearly. Eye seems to have maybe 170,000, I mean 170 million. So the 10 million photonic receptors either died or they were never expressed, right? So the point is, he says, I can't do anything to improve your vision at this time, even if I give you whatever combination of the specs. Anyway, the second thing is the audio as a example which we have taken because we seems to have a little more understanding of the audio process. For optics or for fragrance or for the touch and there are various other sensory stimulus which we can get. We do not seem to have that entire understanding. However, the model which we are evolving, or model the acoustic which we are modeling on, the infrastructure which we are putting together, which in our understanding, I think should be generic enough to take care of others. However, I do not seem to say it's perfectly a good statement. But yeah, that is the underlying understanding I think we carry. I hope I answered you.
No, I think. And the other point is that we started this discussion to understand how brain processes speech.
True.
That is how we started. Right. How brain processes language, and our point was,
right.
It is. I mean, if we go back, we wanted to do a video about story of Sanskrit. But that led us into that first we have to understand, that speech is an endowment. It's not learned. Expression is an endowment. And brain is capable of handling any
Right.
stimulus. And so that's why we focused on speech,
Right.
to just answer your question, Devaji.
So basically, this is a modeling for how do we listen? How does brain
How do
listen?
you speak?
Basically,
How do you speak?
Yeah.
Not that you see, it's not defensive. I'm not becoming,
that is 100. That is right. Premji, thank you.
no, it's not defensiveness of this. The question is, it is not this is right or wrong or this thing. It is a difficult game. So what we are doing is how to simplify it. Can we model it? So the many different anatomical areas we have, they are simultaneously engaged. They are each interrogating, receiving, and hence protein is folding, is protein communicating. So this is happening. The question is, can we tackle it?
Yeah.
So what is the method of tackling it?
So yeah.
So the problem is definitely naturally very complex, and says no, you can't tackle it. That's a very good thought.
Just acoustic interaction. Talking about speech, acoustics, the sound also interacts with this acousto-optic effect. There is acousto-mechanical effect.
Right.
So the thing that acoustics is simple is not really correct.
No, not simple. They're action oriented.
Right.
So what I'm trying to say
Right,
that
right.
acoustics, if you include, basically
Right.
acoustic means a vibration within a certain frequency range, right? So
Right. So
that
when the
range,
action takes
the
place,
interaction is not linear.
okay.
Okay.
When the action takes
So
place,
the brain may be when acoustic signal comes to the brain, it may be exciting many other channels. For example, right now, I started with the thing that what stimulus my brain got to tell me that I missed a very important event, supposed to be at 9 o'clock. Now it is 10 o'clock. So what is that process? Maybe not related to the modern part of it, but thank you.
Okay, any other question? Or Bela, any comment on Prem's presentation?
Bella, unmute yourself.
I would say that the model can be generalized because if we think of that every sensory impulse that conducted envoyed to the brain is in form electrical signal. So either acoustic or visual or odor or taste, the receptors transform this sensory information into electrical signal. So the electrical signal is a common entity, either is auditory, visual, odor, or something. So, so I mean that the brain works with a homogeneous signal, electrical signal, everything. And how it is complex, the connection between or among the different sensory neuronal centers, it means when you ask a question, I hear your voice, but parallel, I see you. So from you, not only just simply auditory impulse, but the visual information is coming to me. So my brain should analyze both and combine these two information that makes one complete visual, complete cognitive understanding what you are, how you looks, and what you are asking.
Right, right. Very good. Yeah,
the
yeah. I think we are so preliminary. I think
electrical,
as Prem said, that the complexity of it. See, the new thing is the following. The new thing is, which we kind of gradually discovering. I think maybe Ramaswamy will comment on it. That is the proteins. So there is a give and take. As I'm speaking, thousands of proteins in your brain, they are folding. So how much of energy it takes for the protein to really fold? And then what happens next? That communication, when it does, so let us say we have identified proteins. What basically, if we are bold, a model can be that for the whole universe information, all information that can come to us, the globality of information, if that information would be channeled in the brain in some marker, protein marker. So that is a question is how much of this processing you can do in a finite amount of time. So it looks to me that could be in thousands, not in millions.
Yeah.
Which means we have thousands of channels, possibly. We have kind of this hypothesis.
I like to connect to what Bela said about the electrical.
Just can I finish? Just
electrical signal.
Let me finish this.
Okay.
So those kind of so those channels, which are the information channels to the brain, whether the protein is connected, see how many facets of red, how many facets of blue, how many textures, how many different of tests, how many different questions of toughness, sourness, love. So that kind of a thing, which are basically connected to the action. So there is some kind of a balance, biological balance, between a digitization of the actions, how many states you can act, and those actions are connected each to the information that you receive. This is the larger design that we think can be handled. But what we have to do is, what we're thinking to do is to see, parameterize this on this N channels. But then we have to see how to distribute this N in different pieces of information. So that's a story we will tell gradually. Yes, you can go. Say your question, then we.
So Bela, I think you made a very important point that all these stimuli and they are sort of creating some electrical state. The electrical state is changing. And that electrical state can be right now conductive, which are signals, electrical signal, or can be capacitive.
Yeah.
By capacitive, I mean capacitance is more related to the distance between two charge. Can be anything. Is the capacitance of a parallel plate is basically epsilon by d. D is the distance between the plate and epsilon is the dielectric constant. The reason I'm saying this is that the dielectric constant is a complex information type of possessing unit, because the dielectric constant is related to the Avogadro's number, and also related to the polarizability of the molecules.
Yeah.
So I think the time has come to go beyond the lumped circuit model of the electrical model of brain, go into more microscopic theory of brain, neuronal function, because what matters here is the shape and the size of the neurons.
Okay.
Not only the chemical elements like the, what is that? All those,
Okay.
what is that? No, I mean, I'm forgetting the names, all those dopamine and all those things. But the thing is that
Right. Move on.
it is a
Move on now.
position of
Let us
the
move on.
molecule.
So I think
And
Ramaswamy.
that could be happening in my brain, for example.
Right.
And that is something very important. Thank you.
We'll take care. Sasadri, any comment? Sasadri, around there? Yeah.
Yeah, Ram.
Can you hear me?
Yes,
Yeah,
Ram.
of course I hear you.
Okay.
We're eager to
that-
hear you.
Yeah, I've been working on the brain architecture
Yeah.
because I worked on hard drives.
Yeah.
Where we are storing the bits and then they turn into bytes and words, and then eventually a computer turns it into a lot of other.
Some of your sound is not coming good.
I don't know what to do with.
I need your comment on this non-linearity of the interrogation. I need your observation.
Can you hear me now?
Yes.
Yeah. Okay.
Yeah.
Non-linearity between the information exchange and how can we handle it? There's a parallel processing going on. So can we linearize it, or any thoughts you may have, like the interaction between memory, hippocampus, neural centers, thalamus, and the brainstem, to how much we can linearize it, and or you say that you take in the following increments being extremely picosecond scale, where you are saying what happened.
Everything happens in small increments, and each step is just a small step goes from one to the next.
But you think like
it happens.
Or it's a microsecond.
No, well,
nanosecond.
The first is going from the analog signal to first creating a digital pulse, and then taking all these digital pulses into sequences and turning them into patterns. So when I started my brain architecture work, from something very small, it just grew on and on into very high level. You have to solve like the mind-body problem before you can tackle the brain architecture. That's really how I have come. And I gave a presentation last month in the Sadhguru Center, and then I had the privilege of talking with Jaspal and others on Wednesday. Let me see how I can summarize it in in a quick fashion. First, you need to understand the difference between mind and body. Okay, and then if you think mind, I think of it as an organ, but it is located inside my skull in the form of a portion of the brain. Okay, and that resides individually inside my cortex, and then the rest of my body has all these physical, sensory, and motor organs, and they have nerves that are connecting back to the mental brain portion of it. And in between that is the midbrain. That is what we are talking about primarily today, which is consisting of the thalamus and the hippocampus and prefrontal cortex. But those are the major the centers where there is relay portion happens. But in between, you have to also have motor commands coming from the motor cortex that's going to separate this, doing the dimensionality reduction or the reduction of the signal into smaller and smaller chunks. So the first smaller chunk happens in the primary sensory cortex. Then the second, that is called the beta process. The second one that happens in the hippocampus, that's the theta time frame. Then in the prefrontal cortex, the third one happens, that's the delta. And so what I have done is develop a concept called open systems, and how develop a mathematical explanations, statistics, and the things like that. So putting it in a one sentence, I'm saying when the information travels from one, from the thalamus to the primary sensory cortex to the hippocampus to the prefrontal cortex, the time it takes follows a very beautiful ratio, which is the golden ratio, that is because everything happens in small steps, and small steps, that is an plus 1 equals an plus an minus 1. That relationship is just a Fibonacci series. And the solution is it's a geometric progression, golden ratio. And if you look at the time it takes to make a chunk of signal, gamma is 25 milliseconds. Then the beta is 65 milliseconds, which is 2.6, which is 1.6 squared. That's 2.6. That's so from 25 to 65, you go to beta. And then 65 times 2.6 is, you get to 175. That is the theta. That's the 6 hertz. And then from there, you get the delta, which is 450 milliseconds. And that, so when we are talking about going from acoustics to an auditory signal, that is, the auditory signal is of a length of a word, about half a second, 450 milliseconds. So what we have to do is we have to look at the whole information pathway and decide what portion of this information pathway in time and spatial scale that we want to deal with. And when we are talking about acoustics to auditory, we are only focused on this midbrain section going from thalamus to prefrontal cortex. I call it the 1 second brain. That is all the discussion we are doing now. It only goes from 10 milliseconds to 1 second. That is how much of the brain we can really talk about in this context.
I'll interrupt you. Like what we want, the question that comes, which will come gradually, is I think we think from the given literature that it is the metastability of the proteins which has different electrical potential and a possibility of configuration that it can twist to come to another potential. And that is where the information is being digested. Stimulus is being digested.
No, that's what I wanted to say.
But the question is how much of electrical field we are talking about, any measurement that you know of.
If you're talking about the verbal language processing, we are dealing with, 1 second, we are dealing with 10 milliseconds to 1 second, and that is too long a time frame to be talking about quantum physics. Okay, so what is important is, it is like trying to understand the structure of my table using an electron microscope. That you don't want to go to quantum scale to understand the second time frame.
Right.
So
no, it is not necessarily human speech and so on. It is auditory sound. That can be volcano here.
No, in fact,
that's okay.
I go even beyond. I'm defining language as an expression of our memory, and in my definition, walking, swimming, painting, sculpting, everything is an expression of our thoughts. Everything is a language, and they can all be broken down into the same concept of words, syllables, and the phonemes type of concept. And the beautiful thing is the midbrain portion that connects my body sensory motor organs to the mental portion, which is actually doing only digital signal processing, the mind portion of it, going from the analog world to the digital portion. That midbrain is using the same neuronal processing, neural syntax. And it's so beautiful that the verbal also follows the same, the neural expression. It really has to, because everything happens in hardware, and you have to understand how the hardware works. And I'm saying the hardware works in this chunking process going from the thalamus to the prefrontal cortex. And that is the heart of language expression. But I'm not limiting it to just verbal communication. The difference between verbal communication and swimming is in the verbal communication, you're using abstract symbol processing. So every change that for every phoneme, for every syllable, we have given it a label. And then for every combination of syllables, a word, we are given it a label. So we are able to communicate just in form of labels, just like a mathematician communicates things with an algebraic equation.
Get it.
We people communicate
let me go
with
to
words.
Sundar Chakravarti, see if he has anything to say. We have to wrap up. Sundar Chakravarti, are you there?
Yes, sir. Thank you very much. I have no specific comments, sir. I listened. I have a lot to digest.
All right.
Thank you.
Very good. So Bharani, why don't you wrap up the comments that you heard, and then how you see the presentation, and then
I actually have a question for Ramaswamy, sir. What is this analog world of which you speak?
What is the analog world? Okay, the sound waves that are coming from the outside. That wave propagation is what I'm talking about as an analog wave. When that wave comes and hits my eardrums, the fluid inside starts to vibrate, and that vibrates the basilar membrane, and the basilar membrane connected with the outer hair, inner hair cells for transduction of the sound, and the outer hair cells to change of the basilar membrane working together. They then produce the electrical spikes that are going through. So the analog, which is continuous energy that is coming into my ear, became discrete pulses, and these pulses happen at 25 milliseconds period, and this is called the gamma, right? So essentially, nothing can happen instantaneously. You need to quantize it. I'm saying in the brain, the interval where everything gets quantized is the gamma period, 25 milliseconds. And then you chunk it into larger and larger intervals, and they're all happening simultaneously in different places. Hippocampus is just one node. The prefrontal cortex is another node. The sensory motor cortex is another node.
Bharani differs from there, but that's okay though. He differs from this logic.
I know. That's why I listen to Bharani also. I'm just giving you my viewpoint.
Okay, that's okay.
Yeah.
No, that's wonderful. That's fine. No, what was confusing was that you had differentiated between the midbrain.
Yes.
And you said that only above the midbrain is digital and below that is analog. That's what kind of threw me.
Right.
Because I thought that it was digital from the cochlea all the way up.
Yeah, from the once it gets into the brain, it is all digital.
Okay.
So I'm putting thalamus into the midbrain.
Right.
So from the cochlea, what goes into the thalamus is the electrical spikes. No, I'm differentiating between the electrical spikes and the pulse processing. I'm talking pulse process. The sequencing is what I'm calling as digital processing. Okay, converting an acoustic energy or a light energy or a pressure energy into an electrical spike. That is still analog version. Right. So the creating up to the electrical spike is analog processing. But once you have it as pulses, that's exactly what we did in digital in the disk drives.
I think again we'll wrap up. But what has happened is we somehow have developed our own group discussion and so on, that the human understanding, the human reception of the signal stimulus is quantitatively different than animal reception. And that we observe by our own empirical, it's that an experiment of this, so which basically boils down to that the quality of the signal, as the signal comes, it is not the quantity of the signal or the character of the signal, but it is the call of the signal. My mom's voice, it is a tiger, it is thunder. So there's a signature to it. That signature gets communicated rather quickly. That's what Bharani. But anyway, you can't argue on this. So this is how the model we have accepted. And then once the brain configures into that kind of a mode, so there are modes of the brain that is communicated by this kind of a signature, so which is rapid, and then the brain tries to decipher what the character and the nature and the context of the signal is, which could be slower and so on, but could be, again, as Prem presented today, memory interaction depends on how many data points you need to say, is this thunder and let go. I know it.
Yeah.
Until a volcano comes, and then you say a little more, oh, okay, do I have to run? Things of that nature. So it's all empirical. It is not really.
No, but no, there is nothing in my opinion, there's nothing really special about human beings. We came out of a single eukaryotic cell,
okay,
and we are just a collection of trillion cells.
No, we're not arguing.
Evolution is the fundamental thing.
We're not
There is a
arguing.
very
We're not
famous
arguing.
quotation. It says, "Nothing in biology makes sense except in the light of evolution." So if you don't follow evolution and don't believe that human beings came out of the same single eukaryotic cell, my monkey ancestor which came from the reptiles, and Paul MacLean's triangle.
What we are saying is, again, I'm not arguing. What we are saying is, that just Paul is not here, we're saying is the speech is an endowment to us, which is not evolutionary. It doesn't happen by evolution.
No, that's one thing. Abstract symbol processing is the pinnacle of human evolution.
That's okay.
Okay, that is what has enabled us to communicate.
That is where the animal and the human is becoming separated, in the sense this whole process of characterizing the universe in syllables is a whole different process that does happen. And where we're going, we do not know.
No.
But it is not Darwinian that I can, and people have made some kind of analysis Darwinian, and that has not worked. So you can invoke anything you like. So, but anyway, so that's not the point here.
No, but animal baby understands the mother-baby relationship is as beautiful as human mother-baby relationship.
Again, when I'm confusing speech versus understanding.
No.
Okay, all right. Bela,
symbolic processing is different.
I think a problem is the animal and the human information processing, visual and noise processing, that we cannot interact, we cannot talk with the animal, what's their experience, how they see the environment. I think that Ram process the visual and the noise information as we process. That the difference is that how we understand how the other partner processing the visual information, if we can ask him or her, what's your experience? What do you think that what is that? The problem is that we cannot communicate the animals. We don't know their communication, because the dogs and the wolf produce vocalized sounds, and they can understand. And the monkeys, they call them together and they assemble in the group, or they escape some danger when the observer at the top of the tree observe a leopard coming, and they vocalize a sound, and everybody knows that there is a danger is coming, so they escape in the group. So of course the cognition that we say that the human is superior because we understand and we use a different communication, the language, as just it is working between us, among us, in the people. If we could understand the animal communication, I think that we would get a different
And also
view.
one point
of-
that Prem put it together today, I think we have to close it, but I just want to mention that the human being becomes individual, customized. So it is not a generic predictability that this will happen, the animal will do. Human may or may not do. So that whole cultural, habitat related
Of course, yes,
protein formation,
yes.
so that's new in the sense that we don't know even how to model it and how many categories exist, but that is happening. Each of our brains are different.
Yes.
And why different? So you can't take a generic this thing that he needs food. So I don't need food. I need dignity. What is dignity? So those kind of things.
Yeah.
So let us now wrap up and say, so Bela, so the next lecture is yours. So I was thinking March 16th as a date. Is it a good date for you? Or shall we discuss later?
Yeah, we discuss later. Yeah.
So tentatively, like, I thought that for the people to know. So something like in March, will
Yeah.
be the next one, and then we shall
Okay.
see.
Yeah. Around.
And the
Yes.
following one. So anyway, so thank you all for participating. As you can, this is okay. This is complex, and Ram has an important point, because that whole field study of literature, we're aware of it and we're trying to deal with it. Most of our parametric information will come from those studies.
Okay.
But something has bothered me as individually and intellectually is this, as Bela just said, the humanness of it. Bela and I, we kind of involved in the human, is a mouse brain we are doing. From mouse brain to come to human brain. Anatomically, they're probably the same. Thalamus, this thing, they exist. But the functioning of it and this nonlinearity part of it. So that is where I think if you can give us some insight that this is how a nonlinearity in the sense, like how do you really, does it become spurts? Does it becomes relays? Does it takes in this thing, forms a grade, and then it responds? So all that we have to model. We are modeling, trying to see.
Yeah.
So thank you all for your contributions and help and attendance in today's lecture. We recorded it, and then it will be available. And next one would be in March. So happy
Thank you.
holidays and happy New Year.
Happy New Year to everyone. Yeah.
Happy New
Thank
Year.
you, Ram.
Bye.
Thank you.